All right, in the last video with micropipettes, I told you how to use the micropipettes as stabbing devices. Oh no, sorry, that was a different video. Oh no, those are for students that want to me when I tell them that they got to redo this experiment over and 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 over again because they're not getting the right data, and that's because they're not using the micropipette the proper way. Don't be that person. Don't use this as a weapon. I should put this on the weapons list of Cape Fear Community College. So, with that said, how do we do this the right way? So that way I don't the person beside of me or the instructor. That might be me, that might be somebody else, and I don't want you to hurt anybody. Okay, so with this operation of a micropipetter, we've already determined the size of micropipetter that I need. Okay, ding ding, a check. Okay, I got my micropipetter, I got it in front, I know which one I want, and then I'm going to choose the right proper size of tip. So the tip goes on to the micropipetter. All right, yeah. So I'm getting another step closer in order to deliver volume the proper way. So now I've dialed it in and I've told you, keep in mind, our big student version of micropipetters, well, there's a 1000 and that's 4 digits, and there's only 3 digits on our little dolly meter. So you want to make sure that you dial it in the appropriate way. So 100 is actually 1000, 060 is actually 600, 010 is actually 100, and that's because that 4th digit is that little bitty tick mark. So I know, it's not very user friendly, but that's what we get for choosing cheap little micropipetters for you to do your training with. So now everything is set up, everything's ready to go. So the first thing that I want to do after I choose the right pipetter, choose the right tip, dial it in the proper way, then the very first thing that I'm going to do is I'm going to go up here to this plunger and I am going to go to the first stop. And the reason that I say that is because as you take your thumb on that plunger up at the very top, look, right here's the plunger, right there. So as I push down, I'm going to get to a point where it's going to resist me a little bit, right? I feel like that is as far down as I can go. And again, I'm not going to give it the Incredible Hulk. I'm just going to kind of take my finger and just slowly push down. And there's going to be a stopping point. At least it will feel like a stopping point. And that is what we call the first stop. So I'm going to go down to the first stop first. And the reason that I'm doing that is because as I push that plunger down, guess what? Air is coming out of the bottom of that micropipette tip. And I don't want to bubble air through my sample. So I want to make sure that I push down first. Number 2, place it in the solution that you want to suck up. So as I hold down that volume, as I hold down that micropipetter, that is great. That's what I should be doing. Keep my thumb on the plunger. I then lower it and submerge it into the solution that I want to suck up or that I want the volume for. Number 3, well, what I'm going to do now is I'm going to slowly I did that sound effect pretty good, didn't I? I don't have it lost yet. Okay, so I'm going to take my solution and I'm going to slowly, and I'm not going to attempt that again because I probably can't do it again, but I'm going to slowly let my thumb up. And the reason that we do it slowly is because if it just kind of goes up into the micropipetter, then I have a chance of getting some of this volume up into the gears and up into the mechanisms of the micropipetter. And you know, today's water, so water can only do so much damage, but what if it was like concentrated acids or bases, or what if it was organic types of solvents that could get in there and really do some damage? I don't want that to happen, right? So I'm going to slowly let my thumb up off the plunger, and as I do that, it's going to suck up into the tip. It should not suck up into the micropipetter. It should only go into the tip. That is it, nothing more. So after that point, when I have my thumb completely off of it, then number 4, I'm going to place it in my receiving container. So when I go to that test tube, or when I go to that beaker, or when I go to that flask, whatever I'm delivering this solution into, I'm going to go to that and I'm going to put the tip of the micropipetter into that container in some form, in some fashion. When it goes into the tip and I'm ready to deliver, then number 5, I'm going to slowly go down to the first stop again. And that is going to get rid of the majority, majority of the solution. Some will still remain. The reason that I say that is because these micropipetters, they are not TD systems. And what that means is that they are not made to deliver. That is what TD stands for. So they are made to contain, not to deliver. That means if any residual liquid is in the micropipetter tips, I need to be able to blow those out. And I'll tell you how we do that in just a second. So I'm going to go down to the first stop. And when I go down to the first stop again, hey, I did it again, didn't I? All right, so when I do that on the first stop, there could be some little bit of remnants, some little bit of remains that's left over of that solution. That's got to get out of there too, and that is the purpose of going further. So actually, after the first stop, I can take my thumb and I can push down and I can go, and I can go down to a second stop, and the purpose of the second stop is that it will take a poof of air, poof, and it will blow out any of the residual volume that is still left over in the tip. So that is the proper way of using a micropipetter or a micropipette. So first stop, and then I'm going to suck solution up. First stop to deliver the majority of that solution out. And then I'm going to go and I'm going to push down a little bit further on that plunger, and that will take it to a second stop, which will use a little puff of air to blow out any remaining residual liquid that might need to be delivered and out of there. That is the proper use, and I will keep in mind, I will say that we are using micro quantities. And the micro quantities, the reason that I'm mentioning that is because this could be a fraction of a drop, folks. These things can be super, super tiny amounts. So ideally, what would happen is that if you are using very, very small quantities of microliters, then it's very often to good measure to touch the sides of the container, to touch the sides of the flask, to do something in order to make sure that you transfer that liquid from the tip into the container. If not, what could happen is that that little bit of residual that is hanging off for dear life on the micropipette tip. So here's my micropipette tip. There it is. And then there's a little bitty squeegee little bitty drop right there on the end, and I'm pushed it out, and when I have pushed it out, that little drop is clinging on for dear life. Don't let go, Jack, don't let go. So it's just hanging on, and it's like, no, please don't make me go into the unknown abyss, into that beaker or into that test tube. And then if I don't actually kind of touch the edge of that tip to that container, it actually might be strong enough to hang on there, and it will never transfer, it will never deliver, it will never go to its intended purpose. So you really do have to let go, Jack. I'm sorry, but knock you off the floating door in the ocean and make you sink to the bottom. That's what we've got to do, and that is the purpose of the second stop, which is a little poof of air that will help blow it out, and then also touching the container sidewall or touching the surface of that liquid just barely in order to make sure that that transfer takes place. All right, so the external video is going to walk you through a little bit of motion, at least, on how to use these micropipettes properly, and just keep those steps in mind as you watch those external videos, because I think visually seeing it in motion, and now that you've heard it from my mouth, as crazy as it is, I think hopefully that will help you out in the lab when you get to that point. Next up is an example problem. I just really want to kind of touch base with this example problem. I'm not going to do the full example problem for you, but I'm going to kind of give you some insight on how to solve this problem. So the class problem here says, suppose a technician is verifying the accuracy of a micropipette. That is something that has to be done, right? We've got to make sure that we are using tools that actually are delivering what they're supposed to deliver. So the technician delivers 500 microliters of water. 500 microliters of water, that is 0.500 milliliters, because they are 3 shifts of a decimal apart from each other. So 1 milliliter equals 1,000 microliters, and that is the proper conversion that we'll do. In other words, what I can say here, off on a tangent, what I can say here is like, oh, okay, so 1 milliliter equals 1,000 microliters. That's good. So this is 500 microliters of solution. So if I want to do proper dimensional analysis, I would do 500 over 1, because that is my given. That is the number that's in the problem, 500 microliters. And then I'm going to bring in a conversion step. Microliter on top, microliter on the bottom, and then we go to milliliter on top. All right, so with that said, what's the relationship? How are they related to each other? Are they brothers? Are they sisters? Are they cousins? Are they mother-in-laws? No, not that type of relationship, silly. We're talking about conversions. So this conversion is 1 mil equals 1,000 microliters. So if I go into this conversion step, down here at the very bottom, there's microliter right there. I see the mu. As far as the mu goes, that's what we call that again. Mu. That is the Greek letter mu. That is not a U. So there is my milliliter, and then here's my milliliter. There's a 1,000 in front, so a 1,000 is going to go there. And then a milliliter, 1 is in front, so there's a milliliter, and 1's going to go in front there. So this is how I do the conversion, and I will take 500, and I will divide by 1,000. Keep in mind, anytime we divide by 1, anytime we multiply by 1, folks, it's not going to change the value at all. So 500, if I want to be prim and proper primrose, if I want to do 500 divided by 1, that's 500. 500 times 1, well, that's 500. And then 500 divided by 1,000, that is 0.5. That is how we do dimensional analysis or conversion, and that's how we get the answer that we do. If you don't like that, move the decimal 3 times. Just make sure that you move it the proper way in the proper direction. All right, so part A says, hey, here are your deliveries. You did 6 of them. Right there they go. So what is the average mass? Folks, do not do this by hand. All right, we got Excel to do it for us. Why not use it? So I'm going to go into Excel and I'm going to enter. I'm going to enter that data in. When I key that data in, then I'm going to go to a new completely clean cell and I'm going to hit equal, and we're going to do AVERAGE 2468. Who do we appreciate? All right, so I'm going to type in the word average and I'm going to open up a parentheses. And then I'm going to take my mouse and I'm going to highlight the data points, and then I'm going to hit enter. So when I do that, it gives me a number, and that number is going to be an average. That's what we want. That's what we got. So hopefully that's going to be correct. Thank you, Excel, for doing the little bit of math that's required in the course for me. As long as I know how to key in AVERAGE and spell it right, it's going to do it. All right, B says, well, what's the standard deviation? All right, well, standard deviation is the idea of how close are these values together. All right, so we want to know how close they are. If I look at these numbers, they are quite varied. I mean, 5.1, 4.9, 5.0, 4.9, 4.8, 5.2. But the answer is like, how bad is bad and how good is good? So standard deviation is going to give me a little bit of that type of information. So when I key in standard deviation, I'm going to do equal, and then I'm going to do STDEV, and then open the parentheses up. Do not use any other version of standard deviation, especially the P version, because P represents population. And you would do that if you had every single data point known to man for every single sample. But we don't really have a population set of data. Most of the time, we are actually utilizing portions of the population, and we are doing handfuls of those sample tests, and we are trying to apply that to the bigger picture. If I did test everything, what's going to go on? So for that reason, I just like for you to use regular, good old, straight up standard deviation straight out of the bottle. So STDEV, open the parentheses up. Do not use any of the other versions that are out there. There are proper times and places for those. But I think to save confusion, if we just streamline it and keep it consistent in our spreadsheets from lab to lab to lab to lab, and only let you worry about that one version of standard deviation that Excel can do, then I think that we're going to be okay. It gets the point across, and sometimes it's all that matters. So STDEV, again, take your mouse, highlight all the data, and then hit enter. And then Excel is going to go and then it's going to give you a number within a fraction of a second, and that is going to be your standard deviation, of course. Next up is part C, and part C says, oh, well, the Micropipette has a specification test that states that it should be delivering a volume of water within 2%. Does the analyst pass, or do they fail in their delivery? All right, well, with this one, we got to talk a little bit more about this whole idea of what a 2% calculation is and a specification test, and does it pass and does it fail, and blah, 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 blah. All right, well, here's the news that I would like to share with you. If you have purchased a very expensive calculator, a scientific calculator, and if you look on the keypad, there's no percentage key. I mean, come on, I've spent a lot of money on this type of stuff, and you're telling me there is no percentage key? And the answer is yeah, that's kind of what I'm telling you. So I'm sorry, you got to do the calculation in a little bit more detail. But you know, if you had a little 4-digit function calculator or whatever, the 4-function calculators, or if you got the calculator on your cell phone, or if you pull up the calculator on your computer, guess what? Percentage is there, and those were free. You didn't have to do anything for them. And they actually make this calculation much faster and much easier. So that's how I'm going to show you first. If you don't like that type of avenue and you do want to be, ooh, look at me, I'm a math whiz, and I'm going to do it on my calculator because I know what to do with percentages, then I'll show you how to do that as well. So let me pull up my calculator and kind of let me show you and describe how to do this calculation fairly easily. Again, using your cell phone. If you've got one, that's not a problem either. All right, so what you see on the computer screen is a calculator, and it says standard up at the top. And the reason it says that is because you do have an option. You can click those 3 little menu lines, and then you can choose a scientific version of a calculator if that's what you want. But I just told you, scientific calculators, they don't have a percent key. So do a standard calculator instead. And if you're doing this on your cell phone, make sure it's standard, because those can also have scientific calculators built in as well. Or if you've got a TI-83 or a TI-84 or anything in between, guess what, folks? You're out of luck. So you're going to have to wait a little bit until we get to that moment when I tell you how to key them into the calculator the proper way. All right, so how do we do the calculator? Why is he going on and on and on about how easy this might be? Well, if you take a look, it says that 500 micrograms should be 0.5 grams. That is the number that we should be getting to, 0.5 grams, right? Okay, so if I am supposed to be getting 0.5 grams, and I do want to say, hold on, horses. This density of water is 1 gram per mil. That's false. Water's density is not 1 gram per milliliter. It is really, really close, but it is not 1.00000. They lie to you in general chemistry. They lie to you in the science courses. This is one of the reasons that when we calculate density and specific gravity, they are often slightly different, because it's not exactly 1. Okay, well, we're just going to pretend that it is, and that's what they do in those classes, and that's what we're going to do for now. But density, it says, is 1 gram per mil. So if we have 500 micrograms, well, that's going to be 0.5 gram is what we should be getting when we deliver 500 microliters or 0.5 milliliters in a container using a micropipetter. So this is actually what we would call the true value. All right, that is the number that we should be getting. Whenever we deliver these, we know there's error. We know there's error in the pipetting. We know there's error with temperature fluctuation. calculations. We know there's error in the balances. We know there's error with me and my technique. So we're not going to probably get 0.500 exactly. And you won't anyway. You're not supposed to, because density is not exactly 1. All right, so either way, what's happening here is that I'm going to take that value, that 0.5 gram, that is what I should be getting, but I will understand that I know that I'll get something a little bit lower or something a little bit higher. It's a matter of how much, right? How much error is present, how much error is accepted, and I don't want it to be too much. So what this is saying is that you've got a specification sheet, and it is saying, hello, I made the Micropipette, and I know that it's working the proper way because we've tested it before we shipped it to you when you bought it. So thank you for your $500 for this Micropipette, and now we're going to send it to you in the mail, and we're going to give you proof, and that proof is a little sheet, and that sheet is going to tell you we tried it out before we sent it. We made sure it delivered the proper way, so it delivered the proper way for us. It will deliver the proper way for you if you use it correctly. That's kind of a specification sheet, right? That's what they're telling you when you purchase it. That is the proof in the pudding when you get it. And it's saying that you should be within 2% of that value. All right, so if I need to be 2% within that value, that means 2% from 0.5. All right, so 0.5 grams is what I should be getting. And that window is plus or minus 2%. 2% of that number. That's what that means. So if I get a regular little video calculator and I'll pull it up on my screen, I'm going to show you how easy this is. Look at this. I'm going to type in, oh, okay. So 0.500, and I need a maximum of the maximum, not max off of Stranger Things, plus 2%, and then I hit equal. Ta-da! That's the answer, folks. That is as far as you can go as far as maximum is concerned. So when I deliver, I know there's wiggle room. I know there's error. But I should not be getting more than 0.510 grams. All right, that is one of the windows, almost like a set point that I should be getting, and I should not get anything greater than that. Okay, now we need to go lower. So I'm going to type in 0.500, and I'm going to go lower. So I'm going to subtract that 2%, and then I'm going to hit equal. So I can go all the way down to 0.49. Okay, so 0.490 grams. Now, if I look at my individual data points, the individual data points might pass and might fail. But we know there's error. We know that sometimes there's more error in some motions than others, and that's why we like to do things more than once. So we're going to repeat it over and over and over. And when we calculate the average, whatever number that average is, we hope that that number fits within this window of opportunity. So I should not be going higher than 0.51. I should not be going lower than 0.49. And it is the average value that we are comparing, because we know sometimes you might be a little sloppy and sometimes you might be spot on. Great job. Good for you. You did excellent. So what we hope here is that your average, after we do it multiple times, fits in that window. So if it does, and again, I did not calculate the average for you, I will let you do that. And if that average fits in that window, then great job. You actually use the Micropipette the proper way, because you were within the specifications of what that laboratory tool was trying to do. So you've been trained properly. Thank you, Tracy. And then you are doing it properly. Thank you, Tracy. Everything is mine, unless it's my fault. I don't want anything to be my fault, but I'll take the credit for everything. All right, now we go to part D, and part D says the precision standard deviation of the micropipette should be 0.01 or less. Does it pass or does it fail? So again, the precision is standard deviation. How close are your darts to the dartboard? That's what that is all about. Okay, so if you do that type of scenario, then the standard deviation can be used, the standard deviation can be calculated. We did that in part B. So I'm going to take that number and I'm going to look to make sure that it is less than 0.010. If it is less than 0.010, then I not only did my job the proper way according to C, if it fits within the window, but I did the same thing every single time, over and over and over and over. Please keep in mind, accuracy is not the same as precision. So I might not be accurate, but I could have done the same mistake over and over and over. So my standard deviation might be good, but my accuracy might be bad. So they do not go hand in hand. One can be good, one can be bad, or vice versa. Now, I want to go back and I want to talk about C again, because there are some of you that just do not like that type of add 2%, minus 2%. I don't know why, but you're going to be maybe some of you that way. So I want to talk a little bit about how do I handle this type of situation, especially if I do have a scientific calculator. So the first thing that you got to keep in mind is that 2% has been derived from a decimal. And anytime we see percentage, that is 100. So a number has been multiplied by a 100 in order to get percent. So to convert this into a decimal, we're kind of working backward here. I'm going to do 2 divided by 100. All right, so if you need a calculator to do that, that's fine. I mean, you can do it in your head too, but 2 divided by 100 is going to give you a decimal place, and that is 0.02. All right, so 0.02. So if you wanted a scientific calculator, if you didn't want to do it on your phone or on a computer calculator and you really wanted to make a math teacher happy, okay, whatever. I'm not a math instructor, but maybe you want to show off and have some bragging rights. Whatever. If that floats your boat, go do it. Well, I'm going to take that 0.02 and I'm going to take the number that I should be getting. So 0.5 is the accepted 0.5 grams. And I'm going to multiply that by 0.02. So when I do that, what's going to happen is that I'm going to take 0.500, multiply by 2%, so 0.02. That will give me the window that I should be adding on and that I should be subtracting from that value in order to figure out what my maximum and what my minimums are. So when I do that calculation, it's going to end up being a 0.01. All right, so that equals 0.01. So now I need to calculate a true max and a true min. So I'm going to take 0.500 and I'm going to add 0.01 to it. And then I'm going to do it again, but this time I'm going to subtract 0.01 from it. And that will give me some ranges. And we hope that our accuracy is in that range. Again, if you want the standard type of calculation, go for it, folks. It's easy. It will save you time. But I can take 0.5 and I can add 0.01. And then when I hit enter, I get 0.51. Folks, look up above. We already did that. We already said 0.510 and we did it much faster and much easier. And then if I do it again, I can do, okay, well, 0.500, and we're going to subtract the 0.01, and I'll hit enter, and that gives me 0.9. Go figure! It's the same answer. It's the same value. That's not an issue. So it's going to give you the same answer. I just think that this is a little bit more involved. There's more math steps here. You got to convert percentage into a decimal. You got to take that decimal, multiply by the value that you're after in order to get the plus or minus wiggle room number. And then you got to add that to it. And then you got to subtract that to it in order to get these proper window ranges. Well, what we did before, again, the only thing that happened here, we didn't have to do that mess. We just had to do 0.500 and add 2% and then hit equal. And that gave me that value. Look how much easier that was. So that's going to be somewhat the best way to handle percentage, at least now, especially if you don't like the math portion. All right, next up is the procedure part. And in the procedure part, we will pick back up in the next video. I want to talk a little bit about some tips and tricks, and then I'm also going to bring up the spreadsheet as well in the next video. So we got one more left. I'm not quite done with you yet, but at least go take a break, you know, go get some chips, go get something to drink, and come back. I'll be waiting. Don't leave me hanging, please.